Hello, good morning students. Today we'll study about blastula. Blastula is a hollow sphere of cells called blastomere surrounding an inner fluid-filled cavity called blastocele formed during early stage of embryonic development in the animal. The blastula of various group of animals differ in form and structure depending upon the variety of factors such as size and amount of distribution of yolk. So when the egg it is fertilized with sperm, it forms zygote. Zygote undergoes cleavage. And after getting cleavage, a ball of cell is formed, which is known as morula. Then the inner cells, they will start rounding off and form a cavity inside, which is known as blastocele. And it changed morula to blastula. So blastula has outer layer of cells known as blastoderm and a cavity known as blastocele in the center. There are following types of blastula in the animals, that is coeloblastula, STEREOBLASTULA, DISCOBLASTULA, BLASTOCYST and PERIBLASTULA. So as I've told you, the cleavage is a very important process which leads to the formation of large number of cells, and ultimately the blastula is formed after the cleavage. So first of all, the coeloblastula. It is a hollow blastula containing large spacious blastocele. The blastocele is filled with fluid containing mucopolysaccharide. So it is of two types. Number one, holoblastic equal cleavage, and second, holoblastic unequal cleavage. So these are the two important cleavages which forms two different types of blastula. In the first case, all the cells, they are of equal size. It is holoblastic equal cleavage. Example is echinoderms and amphibians. Second, unequal cleavage, it is seen in the case of frog. Here, the unequal coeloblastula is formed. The upper cells, they are smaller, known as micromeres, whereas lower cells, they are larger and they are known as macromeres. So, the blastocele is displaced towards the animal pole and multi-layered blastoderm is formed in holoblastic unequal cleavage in frog. Next type of blastula is amphiblastula, a free-swimming larva of certain sponges that is essentially a blastula with small flagellated cells in one hemisphere and large non-flagellated cells in the other. It is formed by two types of blastomeres. One are ciliated, half they are non-ciliated. Example is porphyrin, that is cycon sponge. The anterior half of blastula is formed by flagellated cells, whereas posterior half is formed by large rounded granular cells. So two types of cells, micromeres and macromeres, they are present. Next type of blastula is stereoblastula or solid blastula. This type of blastula is composed of aggregate of large sized and a relatively less number of cells without or with extremely small blastocelic space in the center. So blastocele is very small. Example is nereis, mollusks like gymnophionan, amphibians, certain fishes. So it occurs in variety of insects also. Next is discoblastula. Discoblastula consists of disc-shaped mass of blastomeres overlying a large yolk mass. This blastula is a result of meroblastic discoidal cleavage in most of fishes, reptiles, and birds. There is no blastocyst. Instead, a slit-like cavity called subgerminal cavity appears between the blastoderm and the yolk mass. So, blastodisc is present on one side of the yolk mass. So this is the example of discoblastula. So in this way, the cells they are formed. The blastoderm is formed on one side of the area. Next type is blastocyst. The blastocyst stage is present in mammals. It consists of hollow spherical vesicular blastula containing inner mass of cells at the animal pole. Very important. The embryo proper develops from the inner cell mass. The outer single layer of cell which encloses the blastocyst is known as trophoblast. And this trophoblast, it establishes relation with the uterine wall and help in the nutrition of the developing embryo. Next type of blastula is periblastula or superficial blastula. A stage in the embryonic development of most arthropods have centrolethal eggs. The periblastula is a vesicle whose wall consists of one layer of cells whose cavity is filled with unbroken yolk. It forms as a result of superficial segmentation of the egg. There is a single layer of blastomere around the uncleaved yolky part. Example is insects. So in this way, in the case of insects, the periblastula it is developed. Thanks to www.google.com for providing beautiful pics.